We are Group 14 and today we will presenting about the stages of micropropagation. In our experiment, we focus on the micropropagation process of Zingiber Officinale, which is commonly known as ginger. There are 3 stages that we have done throughout the experiment. First, initiation stage. This stage involves the surface sterilization of the explants to eliminate any microbial contamination. Once sterilized, the explants are transferred into a suitable culture medium to initiate growth. Second, multiplication stage. In this stage, the explants are transferred into a medium containing plant growth regulators, PGRs, that promote the multiplication of shoots. This helps to rapidly increase the number of plantlets. Third, root induction stage. The shoots that have formed are then transferred into a different medium that supports root formation. This allows the development of complete plantlets, which can eventually be acclimatized and transferred to soil.
Next is a method and material. Next step is a sterilization for ginger. Ginger will be selected and then will be cleaned through running water for 30 minutes. While cleaning it through running water, we also added soap to ensure no dirt will stay on the surface. After 30 minutes, the ginger will be put in the glass bottle and will be autoclaved. This is to ensure there is no contamination on the surface of the ginger. Next process is a multiplication. Using 70% ethanol, the surface of the laminar flow will be sterilized first, but make sure that the ethanol does not contact the filter of the laminar flow. Next, the ginger and the media bottle will be put in the laminar flow to make sure the bottle sterilized using ethanol. After that, scalpel will be heat and then using the scalpel, cut the place of the ginger plantlet. Cut the plantlet to ensure it can stand on the agar surface. After the plantlet was put in the media glass, seal it using parafilm and let it stay in the control room in tissue culture lab. Next step is rooting. First, uncap the flask or jar. Carefully remove out the microshoot from the flask. Separate the microshoot into individual propagule. Using forceps, transfer them onto rooting medium, that is MS medium without PGRs. Recap and seal the cap with parafilms. Label the culture. Keep the culture in the control growth room. And last but not least, observe your culture over 2 to 3 days and record your observations.
The result part, for the sterilization part, the explant preparation, we successfully clean it properly using the tap water for the 30 minutes. For the multiplication stage, we cut the sterilized explant into the small section and carefully transfer them into the MS agar medium. We are also successfully doing that. Unfortunately, at the rooting stage, we are not capable to achieve it because the fungal contamination was observed during the multiplication stage. The contamination spread in the agar medium and affected the explant, make it the further development impossible. For the discussion part, why the contamination occur, the main issue in this experiment that we encountered was contamination during the multiplication stage. This is common problem in the plant tissue culture and may have occurred due to incomplete sterilization of the explant. Secondly is airborne fungal spore entering during the transfer. Thirdly is improper aseptic technique while handling the sample. The contamination not only affected the medium but also disrupted the plant tissue ability to grow and develop, preventing successful rooting. To improve in future experiment, we are recommend to extending the sterilization time or using more effective disinfectant. Secondly is minimize exposure time during transfer to reduce contamination. Although our attempt to propagate ginger through tissue culture was unsuccessful due to contamination, this experiment provides a valuable insight into the importance of aseptic technique and explant sterilization. With improvement, future attempt can lead to successful multiplication and root.